Welcome everyone to today's Drug Hunter Flash Talk, and I'm your host, Dennis Koester, Director of Industry Research and Relations here at Drug Hunter. We're happy that you're here today, and we're glad to discuss DILI risk today, drug-induced liver injury risk, with Tran from Amgen. Welcome, Anne.
Morning, Dennis. Good morning, everyone. Oh, good afternoon, good evening, wherever you're joining from.
Actually. Oh, that's right. That's right. Upload the video. So without further ado, I want to introduce our guest, Anne Tran, today. Anne obtained her Ph.D. in organic chemistry from the University of Sydney in Australia in 2014, and followed that up with a postdoc in Jinquan Yu's lab at Scripps. Then Anne started her career in the industry at Arcus Bioscience here in the Bay Area in the East Bay, and significantly contributed to their immuno-oncology portfolio and has actually 2 compounds in phase 2 clinical trials right now. So really amazing achievements in the beginning of Anne's career. She then, in late 2020, joined Amgen in Thousand Oaks, and there she's currently a principal scientist and a medicinal chemistry team leader, and she's working in the space of early drug discovery projects, mainly in the targeted protein degrader space. Anne is also a very active writer. She writes, for example, for the ACS Medicinal Chemistry Reviews, and is also a regular contributor to the ACS Medi division and the GRC Medicinal Chemistry Future Planning efforts. So Anne, really nice to have you today. Welcome to the webinar, and thanks for educating the audience on DILI today. I already learned so much from your book chapter, actually.
Thank you so much for the opportunity. I really appreciate it.
Awesome. Yeah, in the good old Drug Hunter tradition, before we start launching in the presentation, I just wanted to ask you a few icebreaker questions. So what actually inspired you to start a career in drug discovery?
Yeah, I would say, like most people, I do have a personal story of why I went into drug discovery. And for me, it went back to when I was 8 years old. That was when my dad almost had a fatal heart attack. So he survived and thrived today, thanks to really advance in modern medicine, right? So he literally take every possible cardiometabolic drugs you can imagine to survive to this day. And for me and my family, we've been on the receiving ends for so many years. And at that day and time, it really inspired me to be a drug hunter. So one day I can be on the giving end. There's a way to pay forward to the drug hunters who have bring those medicines to save my dad's lives.
Wow, that's a super inspiring story. And yeah, we're glad your dad is with us today. So thanks for sharing that, Anne. What are you most excited about in drug discovery today?
I'm actually really excited about like new screening technology, right? One of my favorite at the moment is the cell painting technology, which is a type of high content and imaging kind of assays that allow us to investigate like the molecular mechanism of multiple organelles at any one time. I'm really excited to see how it can combine with artificial intelligence and machine learning to process those multidimensional data to really advance this new paradigm of drug screening technology.
Cool. No, there's so many like super cool screening technologies out there these days. Yeah, cell painting is actually one really cool one. We looked into that earlier in my career too. So it's super interesting. What's your favorite molecule and why? We love to ask this question because like gets like to so diverse different molecules. So just curious, what is your favorite one?
My favorite one is actually lenacapavir, which is a HIV-1 inhibitor from Gilead, which is the 2022 Drug Hunter Molecule of the Year. Not only because it has a crazy structure, so I think every medicinal chemist loves that structure, right? But my friend from Gilead told me it's nicknamed the unicorn. Affectionately by the Gilead chemists. And I think I was super inspired by the story that was told by John Ling 2 years ago at the GRC. It's really a story of grit, right? It's the perseverance of this compound take 10 years from the discovery to the final approval. It's super, super cool and inspiring story for every drug hunter.
Oh yeah, and I love this molecule, I have to tell you. This is like the once-in-a-half-year injection for HIV. First in class capsid inhibitor. It's like an amazing compound. The structure is absolutely wild. I've heard people from Gilead talking about it. It's an absolutely amazing story. So Very good choice. I can totally relate to that. Next question is, if you could go back to early in your career and you could give yourself some career advice, what do you think it would be?
I actually, so you introduced me as having a PhD in organic chemistry. I would elaborate on that. I have a PhD in medicinal chemistry.
Oh,
Little did I know that earned me the nickname of the Jack of all trades, which is actually equivalent to knowing a little bit of everything and not knowing very well in anything. So I, at the beginning of my career, had fought really hard against this prejudice, right? So if I would go back in time and tell my younger self, is that actually a jack of all trades is not a liability, it's definitely an advantage. And like the unique perspective and voice will definitely be heard and valued. And do not let other people's perceptions define what is possible for you, which is what I did. I limit myself a lot of opportunities because I actually believe in this label, and it's really hindered my career development early on.
Interesting. Yeah, that's kind of surprising, actually. Yeah, but I know, like, medicinal chemistry is PhDs are sometimes perceived weird in the industry, but yeah. So, yeah, thank you for that advice. I think that's good good for our audience to know. So, final question: what do you like most about Drug Hunter?
Yes, I actually been a very early reader of Drug Hunters from the time when most of the content was still free, the super early days, right? So I love almost everything about Drug Hunter, but I have to pick my favorite. I would pick the conference highlight and the monthly pattern highlights. So they not only provide really a lot of information, they also provide it in a very time efficient manner, right? In a 15 minutes coffee time, I would have been able to get myself really up to speed with what's going on in the industry. So very,
Awesome.
very, very, very good job. Thank you for doing that.
Awesome. Yeah, thank you so much. Those are kind of my favorites too. Because for the conference, you can't like find this information anywhere else. And the patent highlights always good to kind of keep up with something. And sometimes it doesn't even hit the primary literature at all, right?
Yeah.
Cool. Thank you, Anne. So before Anne gets started, I quickly wanted to share, because Anne is talking about DILI risk today, and a compound class where this is a particular risk is for BTK inhibitors for MS. So I just wanted to very quickly screen through a case study that we have here on Drug Hunter. So here you can see our one reversible BTK inhibitor for the treatment of MS. You can see the competitive landscape. You can see we're talking about this DILI risk. We're talking about how the target relates to the disease. And for the hit finding and lead optimization, you can find very many transferable insights that you can potentially apply to your project. So, you know, like this has many, many transferable insights, how you can solve common problems in drug discovery, and just, you know, gives you a profile, a preclinical profile and clinical profile of the compound, and also some synthesis highlights that are really interesting about this particular project. So with that, I'll open it up for Anne to share her presentation with us.
Yes.
Yes, perfect. I can see it full screen.
Okay, perfect.
All right.
So hello, everyone. So thank you for joining today. And actually, true to my Jack of all trades label, today I'm going to tell you a story on drug-induced liver injury. From here on in, abbreviated as DILI. Even though it's a toxicology subject, it has high relevance to medicinal chemistry, and I hope I'll be able to convince you about that at the end of the presentation. Sorry, there's problem with the slide transition. First is an outline of today's presentation. We'll go through an introduction on what is DILI. Then we'll delve into the multiple mechanistic drivers that underlie DILI, and also a classification-based method integrating all of these risk factors to really assess DILI risk in the clinic. We'll follow up with a series of case studies, outline successful DILI mitigation strategy. We'll begin to conclude the talk by balancing between DILI risk and therapeutic opportunities in the context of recent FDA approval, and finally with a final conclusion. But before that, so what is DILI? DILI is manifest in the clinic in a really diverse number of ways, as you can see on this diagram here. Probably the most common manifestation of DILI in the clinic is cholestasis, which is the bile flow obstruction, result in bile acid accumulation and subsequent liver injury. DILI can also result in acute liver failure, hepatic necrosis, as well as autoimmune hepatitis. So currently, DILI is being assessed in the clinic through a series of liver injury biomarkers, such as alanine aminotransferase aminotransferase ALT, aspartate aminotransferase AST, or total bilirubin. But as you can see on this diagram here, DILI is a result of a very complex interplay between multiple DILI mechanisms, the patient genetics factors, as well as environmental stimulants, in order to give rise to the complete DILI in the clinic finally. So there is a very strong business case on why we should care about DILI. In general, adverse toxicological findings account for 40% of program termination. And this is a survey that's being carried out across multiple pharmaceutical companies. So with cardiac toxicity being the #1 factor, liver toxicity is a close second. And DILI made up a significant proportion of liver toxicity. Actually, though, in terms of frequency, DILI is quite rare. It occurs in about 2 in 10,000 cases, but despite this low frequency, it gives rise to more than 50% of acute liver failure cases. Because of this low frequency, as well as in many cases, idiosyncratic nature of DILI is actually escape detection until really late stage clinical trial, like phase 2 and 3, where it's already incurred very significant R&D costs. And really the highlight in this case are the BTK inhibitors, where Dennis had really briefly mentioned about that have been put on partial FDA clinical hold. These BTK inhibitors are intended for the treatment of multiple sclerosis. As you can see, these are molecules from different companies representing different chemotypes, and they were put on clinical hold at both phase 2 or phase 3. 3 of these molecules are actually covalent BTK inhibitors. The last molecule, fenebrutinib, who was highlighted by Dennis just then from Genentech, is a reversible BTK inhibitor. All show DILI risk resulting in this partial clinical hold. So as mentioned, the underlying mechanism of DILI is very complex, and this is complicated further by the historically very poor correlation between animal and human pathophysiology. So you see in the subsequent section of this talk, the way in which DILI risk is being currently assessed in the preclinical space is through a suite of in vitro assay and not through animal toxicology study like we would do with cardiac toxicity for an assessment, for an example. So the first and probably most well-characterized risk factor for DILI is inhibition of hepatic transporters. And hepatic transporters regulate the hepatic circulation of bile salts. Bile salts are important not only for the metabolism of xenobiotics, but also critical for the metabolism of nutrients such as lipids. However, because bile salts being detergent-like and also hydrophobic, they actually really cytotoxic. And so their level has to be tightly regulated. Bile salts are being taken up from the blood by transporters such as NTCP and OATP into the hepatocytes, whereby it will be effluxed by a series of transporters expressed on the surface of the canaliculi membrane into the bile. And the key transporter here is bile salt export pump, or BSEP, which regulates the rate-limiting export of bile salts into the bile. So as you can easily imagine, if you have a BSEP inhibitor, you will have a consequence of accumulate bile salts, resulting in liver inflammation and subsequent liver injury. At the moment, there is no consensus on a safe in vitro BSEP inhibition value in order to mitigate DILI risk in the clinic. And that is because, as previously mentioned, DILI is a consequence of multiple risk factors and not just BSEP inhibition alone. So in order to more effectively translate the in vitro BSEP inhibition to an in vivo BSEP disruption risk, the Merck team has developed a 2-tier testing cascade in order to assess this risk. And the tier 1 assay is probably the most well-established BSEP inhibition assay, utilizing an invert membrane vesicle made up of insect cells. And inserting to this membrane vesicle is a human recombinant BSEP. So what this assay tracked is the uptake of a tritiated BSEP substrate, taurocolic acid, or TCA. As you imagine, if you have a BSEP inhibitor, you have a reduced uptake of this substrate into this invert vesicle membrane. This will allow you to determine an IC50 value. And if the IC50 value is less than 5 micromolar, these compounds will then move into a tier 2 assays. It's important to emphasize that these tier 1 assays can only capture the effect of the parent compound in terms of BSEP inhibition in vitro. It does not take into account the effect of metabolites on BSEP inhibition, because this system is not metabolically competent. And this shortcoming is addressed in the tier 2 assay. The tier 2 assay utilizing hepatopac, which is a micropattern co-culture system, between primary human hepatocytes and 3T3 murine fibroblasts. So this co-culture system is effectively mimicking the liver physiology. It also has a really high metabolizing activity and stable phenotype over time. This assay then allows you to determine what the team call a biliary excretion index IC50 value. And if this IC50 value is less than tenfold the maximum unbound concentration at the liver inlet, so there's now a potential for this compound to inhibit BSEP in vivo. So then the project team have to consider to reduce this liability either through an SAR effort, or if they decide to nominate this compound series towards clinical nomination, they have to monitor for cholestasis in the clinic. So I highly recommend you check out this paper from Haffey et al. For the description of this two-tier assay to monitor BSEP inhibition risk in vivo. Another risk factors that go hand in hand with BSEP inhibition is mitochondrial dysfunction. Mitochondrial toxicity accounts for about 40% of hepatotoxicity that relate adverse drug reaction. As you can see and appreciate on this slide, the structure of mitochondrial toxicants are extremely diverse. They also very diverse in terms of their lipophilicity range. They either can be reaching sp2 center or sp3 center. Pretty much there is no way you can structurally de-risk mitochondrial dysfunction risk. And you can also see here that in many cases, these mitochondrial toxicants are also BSEP inhibitors. So if you have compounds that pose both mitochondrial dysfunction risk and BSEP inhibition, there is a very high chance they will exhibit DILI in the clinic. And in fact, many of these compounds are all DILI positive, at least at high dose in the clinic. So there are multiple mechanisms in which mitochondrial dysfunction can take place that you can see here, can either complex 1 inhibitor, electron transport chain inhibitor, or the incorporation of nucleoside drugs into mitochondrial DNA. But regardless of the microscopic mechanism, ultimately what happened in mitochondrial dysfunction is that you will have impaired ATP synthesis because mitochondria is an ATP powerhouse. And another effect is that you have an increase in the production of reactive oxygen species. So currently, the way in which mitochondrial toxicity is being assessed in vitro is through two different high throughput assays. The first one is the cytotoxicity differential IC50 assay, where HepG2 cells are being fed with two different nutrient sources, galactose or glucose. So the main difference between these two nutrient sources is that for cells on galactose, they can only rely on mitochondria to produce ATP. Cells on glucose, however, can utilize alternative pathways such as glycolysis to produce ATP when the mitochondrial function is being disrupted. As a result, galactose fat cell will be way more sensitive to a mitochondrial toxicant compared to a glucose fat cell, giving rise to an IC50 ratio. So if this IC50 ratio is greater than 3, there is a pretty good chance the compound is showing in vitro mitochondrial tox. Another way to assess mitochondrial toxicity is an assay called respiratory screening technology. And this assay utilizing oxygen consumption as a proxy for mitochondrial respiration, utilizing a fluorescent oxygen sensitive probes. So because the fact that mitochondrial toxicity and BSEP inhibition tend to go hand in hand, and when they do, they elevate DILI risk in the clinic, the current best practice right now is to incorporate mitotoxicity screening in really early stage optimization programs, just so we're more informed of the potential DILI risk moving forward. However, to be able to detect mitochondrial toxicity in the clinic is extremely challenging because mitochondrial toxicity is very well known for having a delayed presentation in vivo due to multiple innate compensatory mechanisms. And the best example here has to be fluoridine. Fluoridine was actually is a HBV inhibitor, and it's a mitochondrial toxicant, but it was not detected in clinical trial until the 13 weeks mark, where it resulted in delayed fatal liver failure, ultimately result in 5 patient deaths in late-stage clinical trials. So it's really highlighting the challenge of translating from the early risk assessment to the risk that will eventually be imposed in the clinic. Even though both of the bile salt transporter inhibition as well as mitochondrial dysfunction really offer the medicinal chemist very little opportunity for a structure-based DILI mitigation strategy, reactive metabolite formation gives us a lot of chance because this is really a structure base here. So the overall goal of xenobiotic metabolism is to produce soluble and benign metabolites. But during that process, it also generates electrophilic intermediates that in many cases can be captured by nucleophiles in endogenous protein. So this can result in either direct toxicity or hapten formation that will give rise to immune-mediated DILI injury. We will go into in the next slide. And both mechanisms are operative in DILI. So the current gold standard for detection of reactive metabolite formation is covalent binding study of radio-labeled drugs to primary human hepatocytes. However, as you can easily appreciate, access to radio-labeled drugs is restricted. And so this process only applicable for late-stage optimization program. However, advancing MSMS instrumentation over the years have meant that right now, many companies are shifting reactive metabolite detection to an in vitro MetID format of non-radio labeled compounds incubate with either HLM or primary human hepatocytes in the presence of soft nucleophiles, so that's cysteine and GSH, or hard nucleophile like semicarbazole and potassium cyanide. And so you're looking for the trapping adducts with these nucleophiles. But it's important to emphasize that while this technique is great, it's so sensitive that in many cases you will detect trapping adducts, and the presence of trapping adducts does not correlate itself with the risk for DILI in the clinic. It's just provide a flag. And so you have to evaluate with other risk factors at the same time. You cannot use the presence of trapping adducts as a go or no go decision for chemotypes in terms of evaluation of overall risk in the clinic. Sometimes it can provide a good prediction of DILI risk, and that is the case of Fasiklyfam, for an example. Fasiklyfam generate a GSH adduct, and it was positive in the GSH trapping assay, also shown to be DILI positive. That's result from the formation of the acyl glucuronide then being trapped with GSH to generate this adduct here. Sometimes, though, having no adduct formation does not mean that you are safe. That's the case of Tran, whereby there was no adduct formation at all with GSH or any other nucleophile. However, this compound was shown to be DILI positive, possibly by an immune-mediated mechanism that's still not very well understood at this point in time. And the last two examples really highlight the elephant in the room. Both troclitazone and pioglitazone have exactly the same bioactivation mechanism, as they both have the same problematic motif, that is this thiazolidine dione that being formed through a sulfonium ion then being trapped with GSH. However, only troclitazone is DILI positive, and that is because the enormously high dose of troclitazone compared to pioglitazone. So it's really important that you assess DILI risk also in the context of dose, and we'll come back to this point multiple times throughout the presentation. So in recent years, there really have been multiple advances in order to look at reactive metabolite risk beyond just these covalent binding study or trapping adducts. And that's been the work done by the Merck team. So the Merck team has developed both an in vivo and in vitro rat transcriptional signature assay, which they call a bioactivation liver response assay. So here they look for transcriptional activation of a prioritized set of genes in the NERV2 KEAP1 and the NERV1 pathways. So the workflow is described here. So a compound is either dosed in rats at two different doses, low dose and high dose, in a 4-day study, or being treated with the culture of hepatopax cell repeated dosing at 3 different doses for a period of 9 days. So either the liver or hepatopax then being collect their RNA level of the prioritized set of genes in this oxidative stress pathway is then being analyzed and compared with the control level. And then this allowed them to determine what they call a bioactivation liver response assay score, BALRA score. And it was determined that if this BALRA score is greater than 0.2 at a liver exposure index of more than 3, compounds is showing potentially high DILI risk in the clinic. And this kind of BALRA score and liver exposure index, when used together, has a much better predictive ability of differentiating high and low DILI risk compared to those that had been previously considered gold standard before, such as the covalent binding study. Is it because this assay looking at more holistically the bioactivation risk and not just the target formation of the reactive metabolite species that you're looking for? So I really highly recommend checking out these two papers for the in vivo and in vitro BALRA workflow. It's really incredible work. So related to the reactive metabolite mechanism is immune system mediated liver injury. So this mechanism is really rare and complex, but it has gained appreciation in recent years, mostly due to the increased usage of checkpoint inhibitors. So as expected, it involves both the innate and adaptive immune system. So it's been appreciated that in DILI positive patients and DILI negative patients, they differ by the fact that DILI positive patients actually have drug-specific T cells. It also been noticed that there is certain human leukocyte antigen alleles that are present in certain individuals that making them more susceptible to immune-mediated DILI risk. And the pathways go through here. So the initial cascade is kind of similar to the reactive metabolite risk, where you form an adduct between the reactive metabolite and the protein, with it then being digested further into peptide conjugates. This represents itself as an antigen to antigen presenting cell, and this counts as signal 1. There's need to be 2 signals for this cascade to proceed. Signal 2 can come from a variety of sources. In many cases, it comes from the innate immune system. With 2 signals in hand, what will subsequently happen is a maturation of naive CD4 plus and CD8 plus T cell into drug-specific effector T cells. We can now go on and interact with MHC1 molecules on the surface of hepatocytes, and this trigger hepatocyte cell death and liver injury. So this mechanism is pretty relevant for checkpoint inhibitors such as anti-PD-1 and anti-PD-L1, which are increasingly being approved for multiple cancer indications. And the use of these checkpoint inhibitors are associated with increased risk of low-grade and high-grade hepatitis. So it's important for us drug hunters to really appreciate the fact that if we plan to combine our new chemical entities with the checkpoint inhibitor, we have to have a lower predicted human dose than when we just want to use it as a monotherapy in order to really reduce this immune-mediated DILI risk. An important factor to consider is because as this axis of liver injury is being increasingly appreciated, there's been a call for a need for new screening technologies to really detect this risk in vitro, because right now there's not really a good in vitro system. So this has been trigger move towards long-term 3D hepatocyte culture, or a co-culture system that consisting of hepatocytes and dendritic cells to more effectively evaluate this risk in vitro. So as you can imagine, with all of these risk factors happening at any one time, there have been multiple attempts to really incorporate all these risks in one place. And one of the really successful avenues is through the software called DILI-Sim. So DILI-Sim is a quantitative system toxicology model, and it itself is a model that consists of multiple submodels, each submodel representing an individual DILI mechanism, as shown here, whether it's BA transporter inhibition, mitotoxicity, or reactive oxygen species. And these submodels are linked together, and it also incorporates additional factors like population variability, which is, as previously mentioned in the introduction, is very key to manifestation of DILI. And through a PBPK approach, it's predict the hepatic concentration of both the parent as well as the metabolites in hepatocytes. And with that information, it can allow for the calculation of the concentration as well as predicting the timing of the release of liver injury biomarkers, that is ALT, AST, and bilirubin, and the timing of release into the serum. Ultimately, it will allow for the determination of the key mechanistic drivers underlying DILI. So DILI-Sim was a result of a partnership between academia and major pharmaceutical companies. And so it's best to illustrate really this through a case study. And these are the CGRP antagonists intended for the treatment of migraine. So the first generation and the second generation compound, Tocagabant and MK3207 from Merck, was discontinued because they were DILI positive. So with that, these 2 molecules actually have triggered the Merck team to develop both the in vitro and in vivo BA-RIA score that I previously disclosed, actually. But what happened is the Simulation Plus, the company who developed DILI-Sim, also put in the ALT and AST value into this model as well. And they also come and predict that these 2 molecules are actually predict to be DILI positive if you're using this workflow. And they also predict that Urogabant, the second generation compound, is DILI negative. And that's actually true in the clinic, expanding out that it also DILI negative at the same time. So even though it looks like this software is really capable of predicting whether a compound is positive or negative, and you can basically put in the IC50 of each DILI mechanistic assay and predict whether your compound is posing a risk or not. It does have additional room for improvement, because actually it predicts that Tocagabant and MK3207 driving DILI through BA transporter inhibition. And the Merck team demonstrate through their experimental study that that the main factors underlying DILI for these two compounds are actually reactive metabolite formation. So there's room for improvement. This final step in analysis of mechanism is not quite accurate for these two compounds, but it does have the ability to predict the final outcome pretty well. And I'm sure there will be improvement over the years for this software as well, as people increasingly use it for in understanding potential DILI risk translating into the clinic. Another way of incorporating DILI risk is through a classification-based method. And this is through work from multiple companies. And the first one is GSK and Merck combination. So in 2004, Merck published a paper where they underlined the risk of reactive metabolite and how it's correlated to DILI. And GSK took it one step further and combining this reactive metabolite risk with the daily dose, predicted daily dose, and arrive at a decision tree model, as you can see here. So if a compound has both reactive metabolite risk and it's being dosed as greater than 100 mg per day, there is a pretty high chance that that compound will exhibit DILI risk in the clinic. So you have to consider it for termination. If the dose is less than 100 mg per day, perhaps you can progress the compound to candidate selection with precaution. AstraZeneca took this one step further when they actually introduced a zonal classification of DILI risk here with using two different axes. The x-axis is called covalent binding body burden. You see the unit here, mg per day, because it's actually calibrated for dosed, predicted daily dose. On the y-axis is the performance of compound across 5 DILI mechanistic assay. Obviously, the higher the number, the more in vitro toxic the compounds are. So this allowed them with this 1 mg per day limit for the CVB burden, establish 4 different zones for compound, where zone 1 is no hazard, zone 2 is medium risk, and zone 3 and 4 are high risk for different reasons and probably should be considered for termination. And the current classification of DILI risk we incorporate the work from GSK, AstraZeneca, and we can also put in the recent transcriptional readout work from Merck. So if you have a compound with high transcriptional readout in the NRF2, NRF1 pathway and high VALRA score, and also score in DILI mechanistic assay and also predict to have a high dose, then probably the compound have to be considered for termination. So, but everything have to be considered in the context of the predicted daily dose. So you can see though, with many of these risks have really been focused on the later stage of optimization. But as predicted, there is a lot of interest to move this risk screening to an earlier phase. And that's work done by Pfizer, where they incorporate a machine learning approach using a random forest model. They incorporate the predicted daily dose with the caveat that daily dose prediction at early phase is highly uncertain. But regardless of that, they combine with lipophilicity and ionization class and come up with an IOC AUC score that allows them to really differentiate between highly DILI risk and low DILI risk chemotypes so they can prioritize the early discovery efforts. So I recommend checking this paper out in case of you're interested in a more early phase screening for DILI risk via machine learning approach. So with that, we will switch gears into the final section of this talk. We talked about the DILI mitigation strategy. And the first one is the LPA1 antagonist from BMS. So LPA1 is a key mediator of lung fibrosis, which is the underlying driver for idiopathic pulmonary fibrosis. And BMS 986020 is a first generation compound. It demonstrates proof of concept efficacy in a 6-month phase 2 trial, however, at a really high dose, 600 mg BID. So it came as no surprise that it gave rise to elevated level of ALT and AST with 3 times the upper limit of normal, and it also result in cholestasis in 3 out of 96 patients, with this being the common manifestation of DILI. So with that, BMS voluntarily terminate this compound. However, they've been able to utilize this level of ALT and AST in the clinic and feed them into the DILI-SIM model. And DILI-SIM predict that bile acid transporter inhibition and mitochondrial dysfunction as the key contributing factors to clinical DILI. So using this information, the BMS team were able to set up all these in vitro assays to monitor risk in these 2 particular areas. As you can see, the first generation compound clearly show micromolar inhibition for multiple transporters, not just BSEPT. So clearly represent a risk here. So the way in which they mitigate this risk is to reduce the lipophilicity, as you can see by 1 log unit, increase fraction SP3, but probably most importantly, decrease the maximum daily dose. So it going from 600 mg BID to a final 125 mg BID. in the final compound. And you also see the final compound also stay clear of all the bile acid transporters inhibition capacity. So actually, this 986278 exhibit no hepatobiliary toxicity and is now in phase 3 for IPF. So really highlight the successful DILI mitigation strategy utilizing this suite of in vitro assays and as well as in silico risk analysis with DILI-CYP. The second case study is a BACE1 inhibitor from Janssen. And here they also take a lessons from a failed clinical example, atabasistat. So atabasistat actually terminated in phase 2 due to elevated level of ALT and AST in 11% of patients. This is thought through an immune-mediated DILI. They recognize that one of the key problem motive here is an aminothiazine group that would remove in the second generation into a dihydropyridine. But still, this compound was shown up to be positive in a cyanide trapping assay, which they reason is due to this presence of this dioxane motif here. And through a series of SIR efforts, they were able to replace this 1,4-dioxane with this fluorinated 1,3-dioxylate, which block reactive metabolite formation completely. There was no trapping adduct being detected, since the DILI risk is significantly reduced. This compound was not progressed further, but it was for other reasons of toxicity and not really liver toxicity. So the recommended practice here is try to stick to this rule of 2 as much as possible by lowering the dose of less than 100 mg per day and keeping lipophilicity as low as reasonable, preferably below 3, to really reduce DILI risk. However, as with any risk, we really have to balance with therapeutic opportunities, and nowhere can it be demonstrated as well as this case study here with omaveloxolone. So omaveloxolone is a NRF2 activator. It's approved for the treatment of this really debilitating neurodegenerative disease called Friedreich ataxia. So before the approval of omaveloxolone, there was no approved treatment for this disease indication. And this disease is so devastating that patients only survive up to about 35 years of age. Because of the highly unmet medical need, FDA has exercised regulatory flexibility. So even though omaveloxolone would give rise to elevated liver injury biomarkers, not surprisingly, because it's a NRF2 activator, and you might remember NRF2 is one of the key regulator of oxidative stress. We mentioned it a few slides ago when we talked about the transcriptional signature work from Merck. And so it's not surprising. This is definitely on mechanism that it gives rise to elevated level of ALT and AST. Because of the highly unmet medical need, though, it still went through approval with recommendation from FDA to really test these liver injury biomarkers before and during treatment. However, not all story turn out this well. Vadadustat is a story that doesn't turn out as well. So in 2022, FDA issued a complete response letter to Akebia for this molecule, which is a HIF prolyl hydroxylase inhibitor for the treatment of anemia in chronic kidney disease patients. So the complete response letter was due to increased risk for thromboembolic events as well as DILI. However, vadadustat was already approved in Japan for use in June 2020. And with that, post-marketing safety data generated in Japanese population, Akebia been able to resubmit the information to the FDA to highlight that there's really no DILI risk at all. They haven't seen anything during this post-2 years marketing period in the Japan population that eventually have allowed FDA to approve this compound in March this year. However, this delay in approval of 2 years means vadadustat is no longer the first-in-class HIF prolyl hydroxylase inhibitor in the US market, because in 2023, a similar compound from GSK, because of no DILI risk, was already approved. So it's really in the best interest of company to really mitigate DILI risk to avoid this costly regulatory delay. So with that, I would like to have a conclusion is that DILI, as hopefully you appreciate it by now, arise from a really complex interplay between multiple mechanisms, but the consideration in the preclinical space have to be on a risk and therapeutic benefit analysis. And this case by case, and it's very, very different depending on which therapeutic area you operate in. Of course, if you're in the oncology space, there's more flexibility. If you're in the cardiometabolic space, you are not going to have any flexibility at all, unfortunately. And the successful mitigation strategy so far have been involved in the reduction of maximum daily dose and compound lipophilicity. But it's only possible because these companies have look at the integrating the readouts for multiple DILI risk factors in vitro assays. And there's been multiple recent advancing technologies, such as the hepatopac culture that I talked about, as well as the 3D organoids I did not have the time to talk about today, transcriptomic profiling, as well as in silico risk prediction with DILI-SIM. If anyone interested in these additional development, I really highly recommend check out this two-part ACS webinar that's really highlight very well. And this is actually co-sponsored between the toxicology division and the Medi division. Some of you may have recognized that today's presentation is based on a book chapter that I led the cross-company writing effort of last year in ACS Medicinal Chemistry Reviews. And I would like to take this opportunity to thank all my co-authors who have contributed to the writing effort. I would also like to thank the MCR section editors, Brad and Matt, who truly championed and believed in me, in my ability to deliver on this complex topic at the very beginning. And the two really inspiring and wonderful editors-in-chief, Joanne and Jakob, for their really attention to detail and constructive feedback that really increased the quality of this book chapter. And thank you all for listening today. I'm happy to take any questions, and I'll hand it over to Dennis for hosting the Q&A.
Thank you so much, Anne. That was really, really amazing. I mean, I learned so much about the different assays that you presented for, mitotoxicity. It was really awesome. And we already have a lot of questions in the audience. So for the audience, I see a few raised hands. So if you have any questions, please submit them in the Q&A panel, and we'll get to it. So yeah, let me just start here with some audience questions. We have Ben Taft asking, is BSEP the only transporter linked to DILI risk? I mean, I think you started already talking a little bit about MDR4 and other transporters that may be involved. So that question, I think, is going.
Yeah, I think BSEP, as I mentioned, is a rate limiting transporter, right? But it's definitely not the only one. As you can see from the BMS LPA1 antagonist case, they look at multiple other transporters, right? And it's not just the transporter on the canaliculi membrane, which is where BSEP is, but also the transporter on the basolateral membrane also play a role. So I think while BSEP is the main one, just looking at BSEP alone is probably not a good idea. And I think that's why having the two tiers assay from Merck, the tier 2 assays look more than just BSEP. It's a little bit more holistic assessment. So I think it's also taken into account of some of the other not so well known as well. BSEP definitely the key factor, but I would recommend maybe look at both the tier 1 and the tier 2 assay in a hepatopath culture. I think it gives a much more idea than just the BSEP in vitro assay, like the invert membrane vesicle is actually giving a lot of false positive, which that's not what you want, right?
Right, totally. And I think in that vein, we have another question about the properties of compounds that are typical BSEP inhibitors. So is there anything, maybe any guidelines, like any properties that medicinal chemists can modulate, like a log-D or a pKa that makes compounds more that increases the probability of BSEP inhibition?
Yeah, I don't think there is like a complete structural guideline, but I think there are some association actually. If your compounds is like an OATP substrate transporters, with many acids are, the chance of it being a BSEP inhibitor is also pretty decent, right? So I guess then you should try and minimize like OATP transporter liability, which will also help with BSEP inhibition as well. And I think if anything that resemble a BSEP substrate, right? If you have working in the steroid field, if it's resemble toracolic acid or something, right? But if that is the pharmacophore that you need, then you have to try and see if you can change the structure a little bit to stay away from it. But structure-based study for BSEP inhibition is difficult. I don't want to like sell that idea because I think that's very dangerous. I think the only way where you can really mitigate by structure is in reactive metabolite formation, actually. And with the others, you basically have to set up the assays and put through as many compounds through those assays as possible.
Yeah. I mean, a question that I had was, where in the cascade do you actually start to assess DILI risk, right? Like, is it like, and what do you start with, like, what assays would you kind of start with? Like, let's say you have do you start at the candidate stage or even early and late lead optimization, and kind of what assays would you recommend using in that stage?
Yeah, I think this is actually a difficult question, right? And I think different companies probably have different practice. I guess it depends on where they've been burned by DILI before. for the companies who've been burned by DILI, they probably put it as the early optimization phase, right? Like, no, I don't want to risk that. It was a nightmare for me years ago. So, but I've seen so far that people do put BSAP inhibition and mitotoxicity risk pretty early in like the early optimization phase. And if you start seeing risk, especially in both of those two assays, it's almost a flag that you need to look into it further, right? And I think maybe BSAP, mitotoxicity, and reactive metabolites in terms of like just covalent adduct formation, because it's a pretty standard and not too expensive assay to set up. So maybe with those three, it can inform you with some kind of whether you are in a high risk zone or not. And I guess this question also related to where your target is, right? If your target is in the liver, like GLP-1, for an example, then you are already, by your target being that location, at a higher risk of DILI in general. That if you are in that space, then you obviously have to evaluate this risk way earlier than a program with a target that is expressed elsewhere in the lung or in the heart, right? And I think so it's all very much case by case dependent.
Yeah, makes total sense. Thank you. Here's another question from Hendrik. He's wondering, how do teams interpret MS detections of covalent adducts? Is there a threshold that is commonly used as a flag for DILI risk?
Yeah, this is a difficult question because the problem is it's so sensitive, right? Yeah, I actually heard a talk from Janssen about this. They were like, right now, they literally can see covalent adduct in every possible compound that they put through. And truly, that's not true that every compound has a DILI risk, right? So it's really showing how many false positive that you're going to get. So I don't think there is like a threshold that you can actually use. As I said, it's probably used as a flag. So if you start seeing that, say maybe if you start seeing a lot of those covalent adduct for a particular subseries, perhaps it's now time to consider investing into more elaborate assay, like the covalent binding study with radio label drugs, right? Or if you are willing to utilize like the in vitro BARA score system from to more holistically assess this risk. But I don't think there is like a threshold per se, unfortunately, just because it's so sensitive.
Yeah, makes sense. We had another question from Anonymous. Are there specific liver markers, e.g. Hy's law cases or bilirubin, suggestive of particular mechanisms of DILI?
No, I don't think so. Those are pretty much like a global final readouts, right? So it's not because that you are seeing certain biomarkers that means it will be indicative of certain mechanism. There probably, if there's one, I would say for if you have an immune-mediated DILI, what they have seen, but the problem is this is happening in patients. It's not in animal or anything. In immune-mediated DILI, they tend to see that sometimes for some patients, you just have either drug-specific T cells or the presence of certain HLA alleles, actually. That's only detected in DILI-positive patients versus DILI-negative. That's the only thing that I can think of that in the future, when we have more data, those HLA alleles, there is an attempt to use them as biomarkers to predict the high-risk patient from the low-risk patient, even from like a patient selection standpoint as well.
Yeah. Thank you. And then there's a question about organoids. How do you feel about the potential of them being used, or do you think they're currently overhyped for the assessment of DILI risk?
They are really being used. Actually, I just didn't have time to talk about them. That's why I kind of try to mention it at the end, because I want to pay tribute to that development too. Actually, GSK has already moved their cytotoxicity, because that's another mechanism that I didn't talk about because it's too general, right? So there's of course the compound can just be cytotoxic to hepatocytes, generally cytotoxic. And GSK have moved their cytotoxicity assay for hepatocyte to the 3D hepatocyte culture from the 2D, because it's significantly more sensitive as well as specific. So there's already work in that area. I don't think it's overhyped, to be honest, because I think the problem with DILI is that it's multiple parameters, right? And I think the primary human hepatocyte is really only good for short-term toxicity. The problem when you have something like DILI with so many mechanisms is sometimes you just need time to manifest, because many times need to interact together. So your in vitro system needs to be stable over a long period of time. And actually primary human hepatocyte are not. And that's why you have culture like hepatopac and 3D organoids, which are way more stable over time, that act more like a liver. So as a result, you have a much better chance to capture this risk. A lot of this risk is time related. If you leave these compounds for a longer period of time in vitro, you eventually will detect this risk. But you can't really do that if your system is not stable over time, right? So what you see is actually the system itself is degrading, not the compounds doing anything. And that's really complicate result interpretation.
Yeah, no, that makes total sense. Yeah, thank you. So I guess now we have two more really specific questions on specific programs. So Gagan is wondering if the recently failed BTK inhibitors or halted BTK inhibitors in clinical trials have been looked at in the new generation DILI assays, and what the results of those readouts were. If you,
I am not sure if they have been. I mean, not that I'm aware of through my reading. Actually, the one from Merck KGA is actually terminate completely as well, because so I think the company just deemed it too much risk to take it forward. So I don't think it has been looked at, unfortunately. Maybe if it look at now with the new set of assay, maybe we can detect it way better. But one thing I want to emphasize that for all those 4 compounds that been on, actually 3 out of those 4 compounds that have been on partial clinical hold for BTK inhibitors, they all at pretty high dose. They all more than 100 mg per day. So maybe it's just also that the dose is playing a role here as well. As we mentioned, right, it has to be considered in the context of dose.
Yeah, makes total sense. And then another specific question, this time on Ubrelvy. So you briefly brought up that case study of ubrogepant. And so people are wondering, how did the Merck team dial out the DILI risk in that second generation? I think it's a CGPR.
Yes. It's a CGRP antagonist.
CGRP antagonist, exactly.
So Talcagaban and MK3207, which are the first generation 2.1 compounds, I guess. They both showing DILI positive in the clinic, right? And I think the Merck team has determined that is through reactive metabolite formation. So they use that information and they use that assay cascade to guide all the subsequent compounds and to basically de-risk in that aspect. And Ubrogepant score negative in those in vitro BA-RA score and also in vivo BA-RA. That's allowed them to have a safe compounds towards the end.
Yeah, cool. Yeah, that's always cool to know like different strategies that people have used. And I guess another question towards the properties here is like when considering lipophilicity, like I think you mentioned that Pfizer paper that had multiple inputs, people are wondering about is using log D and log P something useful for a DILI risk, or should people just look at mainly dose?
Hey, I would say look at dose for sure, but like in general, minimizing lipophilicity never hurts, right? I mean, even if it's not helping in DILI risk, it probably helps elsewhere anyway. So I would say consider both at the same time. There is actually some mild correlation. I didn't want to flag that up too much, but there is a mild correlation between lipophilicity and mitochondrial dysfunction. In general, the lipophilic compounds just have a higher risk for mitochondrial dysfunction as well. So there's multiple reasons for trying to keep a reasonable log P or log D. Sometimes it's not possible. It's just because of your target, but try the best to basically keep reasonable lipophilicity and in the context of the dose as well.
Yeah, makes total sense. And then people are wondering about the throughput of these, next generation assays. Are there any ones that are particularly high throughput? I think you mentioned a few in the mitotox assessment. And then people are also wondering about false positives in those assays. Or maybe you can talk a little bit about the high throughput aspect. And then is there a chance to like confirm false positives, I guess, with like orthogonal assays, I'm assuming.
Yeah. So high throughput, I think the mitochondrial toxicity assays are really high throughput. The BSEPs assays are also very high, at least the tier one assay. I think the tier two assay is reasonable. The in vitro BALI score to look at the reactive metabolite formation from Merck. I mean, 2 years ago, it was described as medium throughput. And it's actually done in 96 well plates. Actually, it's already not bad, right? So I think right now they probably have increased the throughput. So I would describe many of these in vitro assays are medium to high throughput. There's not really one that's super low throughput, right? And I'm so glad that someone brought the questions of false positive. Didn't have time to go through that. So I have to emphasize that something in toxicology, but when I start reading, I start to appreciate how different they think compared to how we think as a medicinal chemist in the screening campaign. They're actually not concerned about false positive. They are concerned about false negative. Which is understandable because you assess DILI many times super late stage. Don't want to throw out a good compound. And you would explain if someone asked me why would they not worry about false positive, that is because you have so many different assays, right, to really reassess that risk. You have so many chances to recorrect, even if one assay gives you a wrong false positive result. So minimizing false negative has always been the go-to for any DILI mechanistic assay. And I think they happily trade with the false positive on the equation. So hopefully that answers your question. If you're interested, we have this section in the book that talked about how this is being assessed.
Yeah, interesting. For medicinal chemists, you don't want to give up your compound because it's
you don't want to give up your compound
right?
until the final minute when you can't deny it any further, right? So basically,
been there, done that.
Yeah.
Another question here is associated with the PK. So can you actually minimize DILI risk by BID dosing? So basically, you know, like lowering the dose, but then increasing the frequency of dosing, or like without giving like the medicine without food, with or without.
I actually haven't seen the food effect or the dosing. I'm not sure if people actually have played around with that, but overall, I have seen that it seems that the overall cumulative effect. I don't think you can play that trick, unfortunately, because again, it is a mechanism that takes time to develop. So I don't think playing around with the dosing schedule will actually help that much. Eventually you really have to solve the problem at its core. Is your compound just to have to be so much better PK wise and reduce the dose.
Yeah, makes sense. And then I think you're going to like this question since you're a fan of cell painting. Do you think cell painting and high content imaging has a role in predicting and understanding DILI?
That's very interesting, actually. I'm going to be excited to see. I think it does have the chance to move it in that direction for sure. I think so far it's mainly been used for like in the screening stage, right? But I will not be surprised in the future, especially with all AI and machine learning can help us understanding so much what these morphological change will be. Actually, that's a very good point because a lot of time you cannot really see through histopathology DILI manifestation, actually, even in animal model, right? So I think there's a really good potential there. So
nice. Yeah, we'll look forward to seeing that the data from
that.
Right?
I know.
Nice. Another property that people are wondering about is you know, besides lipophilicity, et cetera, is fraction sp3. Is there any, you know, correlation that has been seen with fraction sp3 and DILI risk, or, you know, nothing that comes?
Yeah, there's some mild correlation, right? Again, there's really never been any strong correlation with DILI, but I think it does trend in the positive direction. So if you increase your fraction sp3, you also decrease your DILI risk to a certain extent. And that's been the case for the LPA1 antagonists as well, where they increase fraction sp3. So it's definitely helping the same way with lymphophilicity. Probably a little bit less, but if you can do so, it's probably helpful.
Yeah, I mean, you showed this great slide with the structural diversity of the mitotox, right? And there was really nothing.
There was really nothing. Yeah,
sp3, like everything was kind of could be
all over the place, right?
Yeah, makes total sense. I guess we have one more minute. We have a lot more questions. So, Anne, like really great presentation. So maybe we'll do one more to wrap it up. And then the other questions, we might share answers online later. But really, it speaks to what how great this presentation was. And so thank you so much. One more last question I'll ask is, could route of administration make a big difference in DILI tox profile?
It's an interesting question. Actually, all of the DILI positive compounds I come across so far are orally dosed.
Oral, right?
Actually, yeah, I have to get back to this question. I don't know the answer to this question on the top of my head, but I'll look into it. I'll get back to it.
Awesome. Yeah, no worries about this. Yeah, and so thank you so much. We're on the top of the hour now. It was really, really amazing. And thank you also for being here to answering all the audience questions. I mean, look, you know, you rarely get the opportunity to talk to a medicinal chemist expert in DILI, and you're one of the few ones out there. So we really appreciate you and you being here, giving that presentation and answering all the audience questions. Thank you so much, Anne.
Thank you so much for hosting, and thank you everyone for joining today.
Yeah, thanks everyone for joining, and you know, you'll catch us at the next event. Thank you so much, everyone. We look forward to seeing you. Bye-bye.
Bye.